Hello, I am Ashley Ford Versypt. I'm an associate professor at Oklahoma State University. My presentation is titled Multiscale Lung Tissue Simulator for SARS-CoV-2 Infection and Damage. And this work is on behalf of myself, Amin al-Islam, and the SARS-CoV-2 Tissue Simulation Coalition. The members of my lab who've been working on the project described here, postdoc Amin al-Islam, undergraduate research scholars Blake Bartlett and Kate Lane. This work is funded through the NIH R35 MIRA, Goldwater and Lou Wentz Foundation scholarships to Blake, and the Oklahoma Center for Respiratory and Infectious Diseases also provides some support. COVID-19 is all around us. We have seen big implications of this. The tissue damage is due to the infection of the SARS-CoV-2 virus and acute inflammation. When the virus interacts with the cells in the human body, it binds through a receptor called angiotensin converting enzyme 2 receptors. Cells that express this receptor line the human airways and other tissues in our bodies. This image is showing a snapshot of a human alveolus, the air sacs of our lungs, and the close connection with the human vasculature, the capillaries, where there's gas exchange. And in a healthy lung, this is an air sac, and this is the interstitium, and then this is the blood vessel. In acute respiratory distress, there's more permeability in the vasculature, there's damaged and permeable epithelium of the lung airways, and there's also can be fluid filling these airways in a phenomenon called edema, and a lot more inflammation and immune cells present here. COVID-19 is a multi-scale problem. There are interactions at the subcellular level where the virus binds to different receptors, or on the surface, endocytosis moves into the cells, replication, etc., signaling responses. At the cellular level, the infected cells can die, but can also secrete interferons to warn other cells. Immune cells can phagocytose dead cells. Infected cells can be attacked by other immune cells. At the tissue level, there's a spread of the virus through the tissue and mucus. Infected regions grow, the tissue damage spreads, for example, in edema, and immune cells coordinate with the secreted factors. And at the systems level, there can be signals to the lymphatic system, the entire systemic immune response, more immune cells arriving at the site of the infection, vascular damage, and a host of other things that we're still discovering. This is a complex problem. So we want to use multi-scale mathematical modeling and chemical engineering concepts to understand the system and find better treatments. I'm going to talk about work that comes specifically from my lab connected with 3 other research endeavors. So the first is with Dr. Kathy Froman, the University of Delaware. Secondly, I'll briefly mention work with Dr. Vincenzo Cifarelli and Dr. Princess Amakwade at Washington University of St. Louis. And then finally, I'll talk about work with the Tissue Simulation Coalition and the people that I work with most closely, Paul Macklin, Thomas Hillen, and Amber Smith. So with Dr. Froman, Blake and I have been working on computer modeling of aerosol particle transport through the lung mucosa, and Blake has this presentation in the undergraduate national paper competition as part of the annual student conference. Briefly, I'll just mention that Blake's work has focused in the last 2 years on modeling the transport properties and processes through the lung mucosa involving diffusion, mucosal clearance, which is a velocity term, and penetration of the tissue. And so we can apply concentrations at some interface and see the combination of transport effects through nanoparticles and the mucus. And so this simulation zone is representing different layers of the mucus. And so now working with Kathy, we're looking at if we simulate where the virus penetrates, if there's a prophylactic or other material already in the mucus, how can those bind together and what's the transport properties of the bound complexes in a way to try to prevent the penetration of the virus into the human airways or other types of clinical therapeutic types of measures. Kate Lane is working on modeling the fluid dynamics, fluid mechanics of edema formation. That's where the fluid is actually penetrating into the interstitium and then into the airways. And so we're looking at this interface between the lung epithelium, the vascular endothelium, and normally there's no flux between either of those cell layers, but there is lymphatic drainage from the interstitium. In COVID-19, the epithelium can be damaged and the endothelium of the vasculature can become more permeable. And so that the flow rates of these different processes are going to lead to the rate of edema formation. Additionally, we have previous work where we have looked a lot at the renin-angiotensin system systemically and in certain tissues of the body. And the ACE2 receptor is actually an important molecule, important receptor for the renin-angiotensin system, which controls blood pressure and other inflammatory disease mechanisms, other processes in our bodies. And so we're looking at connecting the binding of SARS-CoV-2 to ACE2, how that interacts with the renin-angiotensin system, and how that gives rise to different processes in acute lung injury. And with that, we're collaborating with Dr. Cifarelli and Imakade related to the vascular biology, specifically the vascular leakage, coagulation, and inflammation processes. The larger collaborative team that we're working with is known as the SARS-CoV-2 Tissue Simulation Coalition. And this is the title information and author information for our most recent version of bioRxiv preprint. This has many authors on it, and I've highlighted in yellow the engineers on the team. And Dr. Islam is the postdoc in my lab. I'll show you some more details of this work, but if you're interested in following along with the bioRxiv papers or the forthcoming manuscript or the simulation environments, I encourage you to check out our website, which is covid-19.physicell.org. And Physicell is the multiscale simulation platform that Paul Macklin and his team have developed. And so we're maintaining the coalition on their website. So Paul Macklin led putting together this team. And so in just a couple of days in March and 12 hours total, Paul prototyped the first version of this model and his team put it on NanoHub, a web platform for doing GUI-based online computing. And then within the next 7 days, the team coalesced and we a teleconference. We wrote the first preprint and submitted it to bioRxiv. And so since then, we've been working ever since, meeting on a weekly basis since late March. Currently, there are 40 plus regular contributors from 20 plus institutions in at least 3 countries. So we're looking at in-host viral infection dynamics and the tissue level immune responses and other sorts of tissue damage and healing mechanisms. And so this conceptual framework is the epithelial layers of the lung. And so we call it T for tissue. So the circles represent epithelial cells. The virions can move through this tissue, virus cells, endocytose and replicate and have other viral processes inside of the cells, the epithelial cells. Virions are secreted, they can move. Other cells can also move and chemotax through this tissue in response to different gradients, different signals. We have now the immune response that involves various types of immune cells, the macrophages and the neutrophils and CD8 plus T cells. And we also have cytokine and inflammatory chemokines in there. Debris we consider as when the cells have died. So dead cells and then being eliminated and leaving a blank space behind. So we can see if we have no immune response, the blue turn to gray and black and then turn to white. The white is no longer there, the debris. If we have the immune system in the model, we can actually slow down the rate of forming these white areas. And we're working on the simulation with different types of responses, but right now we have ACE2 receptor binding and trafficking, viral replication, the cell death response, rules for the immune cells, as well as immune cell recruitment. And in the microenvironment, the viruses can spread, the chemokine is secreted, cytokines can move around, pro-inflammatory cytokines. We also can consider situations, if we let this go long enough, where with the different T cell populations, we start to see these infected cells, then the immune response is coming in. But if the immune response is too strong, we see drastic interactions over time where the virus keeps spreading, the macrophages are trying to eat the cells or eat the virus up, the CD4 plus and CD8 plus T cells are trying to kill off the infected cells, and eventually the red CD8 plus T cells are actually destroying any cell that had been infected because the virus never got under control. And this is a completely worst case scenario, that all of the epithelium is completely destroyed. We don't think that's realistic, or certainly that would be a very, very bad outcome. There are various immune responses that are possible. No immune response, and immune response keeping things under control by changing the rates of different processes, the kinetics, the binding, all of these different phenomena that the model seeks to capture. Interferons is a certain area that chemical engineer Jason Shoemaker from Pittsburgh is working on in his lab. And so with the interferons that are secreted, depending on the rate of interferon secretion, you could have maintained the integrity of the entire tissue through that interferon signaling that allows the host cells to fight off the infection before the overall immune response happens. So different levels lead to different remaining tissue. My team, we've specifically added fibroblasts, and these are useful for healing and the fibrosis processes involved in later stages of acute lung injury. And so here we've represented the fibroblast in orange, and the way that we have this implemented in our agent-based model framework is that fibroblasts are recruited when CD8 plus T cells kill off infected epithelial cells. We then have a secretion of an anti-inflammatory signal and just TGF-beta, and then the fibroblasts hone toward that TGF-beta concentration and deposit collagen. And so the resulting collagen, this is filling in those white spaces and trying to reform and heal the tissue. Now, this can get dysregulated in inflammation where there's too much fibrosis, and scarring can happen, but that's much better than the situation of completely destroying the entire tissue that some of our other simulations have shown. And this work is all ongoing. And so with that, I have introduced you to our multi-scale lung tissue simulator and some of the other simulation things we're doing with chemical engineering type frameworks with collaborators from chemical and biomedical engineering and other fields. And if you have any questions, feel free to reach out to me.